and patient. So I think for me, at least, it's been very transformative, and I learned a lot. On a personal note, so I don't have kids, and I can only imagine what you are going through as parents in terms of concern, worries, fear about the health of your kids and their future. What I've seen so far is like loving and caring parents that have had heart to understand what their kids go through. And for me, this is very inspirational. And on the last note, I have also the chance to meet several kids today and yesterday. And I have been impressed about how caring, about friendly they've been. They took my hands, they brought me to their family, they introduced me to them. It was, you cannot imagine how inspiring that was. For me, as a shy, introvert scientist who cannot relate, who have a very hard time to actually make connection to other humans, these kids were so impressive to me, and just for that, it's been a fantastic weekend so far. So let me not stop being emotional, because this is not about me, but that's about you. And I'm gonna show you a bit about what we do in my lab, and the role of POGZ, both in the White-Sutton syndrome and hopefully in other type of pathology, that we hope that we can draw funding and hopefully also understand better what POGZ does. So here's, I don't know if you have a pointer, yes. So here are all my information. So here's my email. If you have any question, please feel free to email me. If you see me today or tomorrow also, please come say hi, and I'll be happy to answer any of your questions. So what we do in my lab is primarily looking at the nucleus and a molecule that some of my colleagues have already introduced you, called DNA. What is very important for DNA is that when cells divide, it needs to be replicated properly so no damage or problem are propagated to other cells. And this is a very challenging problem. For one reason is because DNA is a very unstable molecule per se, so making sure that it's propagated properly is very complex. But the other problem is that our genome is constantly exposed to both endogenous and exogenous sources of DNA damage. And for instance, us cells per day can encounter between 10,000 to 1 million different types of DNA damage, which make it the integrity of that molecule very complex and challenging to actually organize. And so this is a series of different types of DNA damage that can occur in the cells. It can be very simple, like just the base that is damaged, but it can also be more complex where the DNA can break into pieces, and that is obviously more complicated to repair. And so for each type of DNA lesion, the cells have actually developed a very complex response to signal, repair, and ultimately resolve those type of damage. And part of the damage that we are very interested in is called DNA double-strand break, and there's two major pathways that are involved in this process called NHCJ and HR. Homologous recombination is probably one of the most famous pathways that you may know of because of mutation in BRCA1 and BRCA2 that predispose to breast and ovarian cancer, and that's part of what we've been interested in to characterize. These are some of the sources of DNA damage that can occur in the cells. So some of what we're interested in my lab is obviously radiation and chemical that are used to treat patients with cancers. But actually, most of the DNA damage that occur in the cells come from normal processes, like replicating DNA or having the cells dividing into different daughter cells. There's also certain type of damage that comes as natural damage or genetically diversifying damage, like meiosis or during B and T cell development. That's also some of the things that we're very interested in my lab. This is a very complex picture, and I don't expect you to gather anything out of it, really. The only thing that you may need to know is that when there's a break, there's a very complex set of proteins that go to the damage to signal the damage and ultimately decide whether he wants to repair it one way or the other. He can repair it in what we call an end-fails-full manner. The idea is just that he will take the two ends, stick it together, and decide that it's repaired. The problem of that strategy is that it can introduce mutation that's genetically unstable, so it's not necessarily the best way to repair damage. There's another pathway, as I mentioned, HR. This is a more complicated pathway, but the idea there is that it's more faithful, and he makes sure that what he actually repairs is representative of the original damage or original DNA that is present there. The beauty of this process is that we can visualize it in the cells. So you can see that normally, this is a nucleus of the cells. You don't see much happening there. However, when you create those damages, you can see the very nice foci. And this is actually all these proteins that accumulate at site of damage that we can visualize in the cells. This is kind of very complex representation of all those pathways. As I mentioned to you, I think the most important for you in the context of POGZ is this pathway called homologous recombination that we think is very accurate and typically repair DNA double-strand break. So some of the questions we ask in my lab is to understand how those breaks are repaired. And there's different level of complexity that influence which pathway is important to repair it. The first thing is whether the break is very complex or simple. So if it's simple, it's obviously very simple to repair it because you can just stick the two ends together. The two ends together, and that happens very simply. When it's complex, you sometimes need to process those breaks, and that makes it more complicated to repair it, and that's kind of one element that decides which pathway that can be used to be repairing it. The other element that actually been introduced by one of my colleagues is the state of the chromatin. So as been mentioned, DNA can be either in an open state that allows genes to be expressed, or we call that achromatin, but it can also be in a very compact state, like we call heterochromatin. And so depending on whether the breaks happen in one context or the other, the pathway that will be involved in the repair may also be influencing that process. And there's other elements like cell cycle positioning that also influence which pathway repair that. And so part of the project that we've been working on is to understand how the chromatin state is influencing the pathway that is important for repair. And in that regard, one of the key proteins that is important for determining whether the protein is in, the chromatin is in heterochromatin state, is called HP1, which is a very well-known partner of POGZ. And when HP1 binds to chromatin, it actually makes it in a closed state or heterochromatin state, while when HP1 is removed, you can open the chromatin. And in fact, what we see is that when the double-strand break is generated, heterochromatin transforms into achromatin, and that allows the breaks to be signaled and ultimately repair, and then allow the DNA repair factor to be recruited. So part of the project that we started with has been conducted by a former PhD student, John East, that spent about 6 years of his life working on POGZ. So just for you to estimate the time and investment of that person to characterize POGZ. And so one approach we used is kind of, I would say, compare it to a paintball. And the idea is that we are interested in one protein and to understand which any other protein come in close proximity. The concept is that the closer my target is, the more likely I will hit the target, and I can quantify how close that target is by the fact that I can hit faster or more that target or not. And that's kind of the idea here. We actually have a protein called HP1, and we want to know which protein come in close proximity to that other protein, and we can then analyze that by mass spectrometry. And so we did this analysis, and in short, one of the key proteins that actually interact with HP1 protein are POGZ, as been previously shown. And what was interesting is that it's not only in a normal state that we see this interaction, but also when we cause DNA damage using a DNA damaging agent called neocarcinostatin. And so the dot, or the size of the dot, corresponds actually to the amount of time that this target has been hit in our assay. So then we kind of wonder what POGZ could do in this context. We know that POGZ is a very big protein that has an HP1 binding site. Several works from a previous group have shown that POGZ is very important in chromatin organization and gene regulation, particularly during neuronal development, but also hematopoiesis. But there's also an emerging role of POGZ as an important modulator of genome stability. And there's some work that has been shown that POGZ is very important in regulating certain processes like mitosis. So the question that we ask ourselves is, can POGZ participate in the repair of DNA in certain contexts? And so for that, what we did is that we depleted POGZ from different normal and cancer cells using different strategy, siRNA or CRISPR. And so using an assay called the COMET assay, we can actually measure the amount of DNA damage in the cells. The idea is that, like a comet, debris can get out of the nucleus and can be measured by gel. And I'm not sure you can see it very well, but you can see that this is a nucleus of the cells, and at the end of this nucleus, you kind of have a tray or an element. That's kind of all the debris that comes out of the cells. So we can quantify these type of debris that get out of the cells, and that's kind of a readout of the amount of DNA damage that you have in the cells. And so what we saw is that in normal cells, we have very limited type of DNA damage. When you actually expose them to ionizing radiation, you can see that this tail increased very significantly, and ultimately, it gets repaired. That's the normal context. Now, what happened when we remove POGZ from those cells? Well, the first thing that we observe is that at steady state without damage, there is actually increased damage in those cells, suggesting that POGZ-depleted cell may have naturally more damage to the DNA than control cells. We also look at whether those damage are resolved over time, and what we also observe is that there's a delay in those cells to actually repair the lesion that we created in that case. And this is the case in two different setups where we depleted POGZ at different levels, where we see the same thing, which is an increase of the endogenous DNA damage, but also a problem in the resolution of DNA repair. So that suggests that POGZ may be important for DNA repair. The other thing we look at is whether POGZ can go to site of damage. And so for that, we can do what we call laser stripe microradiation. So it's actually a laser that we use to generate a lesion in the genome. And you can see that the protein POGZ here can actually get recruited to that lesion very early on during time, and then at later time point, it actually disappears, suggesting that it may be important for DNA repair. We also look at which pathway is actually regulating. And for that, we can actually use a system where we can look at the recruitment of DNA repair protein at site of damage and quantify it. So what we saw as a nutshell actually is that when you deplete POGZ, you impair the recruitment of this very key complex at site of damage, and that impair homologous recombination. And in fact, we can also measure that in a different assay where cells can become green when we repair them. And in fact, what we saw is that cells that have POGZ depletion do have a defect in repairing DNA double-strand break in different type of cell line. Let me go quickly on those ones. So that's kind of a very quick summary of what we've seen in those cells. I think the first thing we saw is that HP1 has a key partner as POGZ, and that POGZ is important for DNA repair, particularly in homologous recombination. And some of the mechanistic insight is that it actually influenced the recruitment of BRCA1. But the question we were very interested is to see what happened in vivo using mice. And that's where some of the comment that you actually been raised about our mouse model is recapitulating what patient has actually experienced was very informative, because we have seen actually the same phenotype that my colleague actually saw, which is a lack of anxiety or less anxiety in those mice. And I'll go through the data and then we can discuss hopefully later on. And obviously, the interest in POGZ rose from the fact that at the time that we worked on it, several reports have identified heterozygous mutation in POGZ, which resulted with this neurocognitive disorder later on called the White-Sutton syndrome. And I'm not going to go through all the characterization, but I think some of the key elements also that we're very interested in certain severe cases is the fact that the White-Sutton syndrome will also linked to recurrent infection, which is an element that I don't think a lot of people have looked at so far, and we've been very interested to look into in the future. So what we did is that we generated a mouse model of POGZ using CRISPR, where we deleted 2 different exons of POGZ. As mentioned previously, POGZ knockout mice are embryonically lethal, so we cannot generate a full knockout mice. And we think that the embryonic lethality comes very early on during the development at embryonic age 14.5, where we start losing Mendelian distribution. But in fact, what happened in patients is actually most patients only have 1 copy of their gene that is actually mutated or inactivated. So we did actually use an heterozygous setup where only 1 allele of POGZ is inactivated and try to start characterizing those mice. And as you would see in patients, those mice typically have a smaller, typically smaller, so the overall weight is reduced compared to wild-type mice. And this is not just an overall body weight problem, but also an organ-based problem where we can see that each different organ is typically smaller, including the brain, which would kind of go with the microcephaly that is typically observed in a patient with the White-Sutton syndrome. Then we try to see whether we can actually recapitulate some of the key features in terms of hyperactivity, anxiety, or behavior in those mice. We're not neurobiologists, so we actually used the expertise of others to do that. And so we also used the open field maze as a way to actually characterize the neurocognitive impairment of those mice. And as you can see, most of the wild-type mice typically hang out in the corner of those maze that show kind of the anxiety, the scariness, while you can see that the POGZ heterozygous mice are kind of all over the place and much more open to actually explore different areas of the maze. And that is typically a way for us to monitor anxiety or friendly behavior as the way of being out of the corner of those mice. Those assays also allow us to actually measure the distance that those mice and the speed of those mice in these assays, and you can typically see that the heterozygous mice are typically more hyperactive, which would correlate with what typically patients with White-Sutton are, as well as less anxious because they are typically more in the open field thing. So our hypothesis is that patients with White-Sutton syndrome are not just developing because of the defect or the problem of POGZ in transcription regulation and gene expression, but maybe also because there's a defect in DNA repair. And so what you could keep in mind is that when cells have DNA damage, they typically stop growing, and therefore there may be a developmental problem because of that. And that's what we're thinking that may happen in this context. So the first thing we did is to see whether our mice is actually hypersensitive to DNA damage. So we exposed actually our mice to a lethal dose of ionizing radiation, and you can see that POGZ heterozygous mice are typically dying faster than wild-type mice when you expose them to that, suggesting that there is a defect in DNA repair. The other thing we look at is whether when we took mouse embryonic fibroblasts, do those mice hypersensitive to an DNA damaging agent, and we also do see that. And the final thing that we also looked at is whether those mice actually display DNA damage in their brain. So we took section of the cortex, stained them for a marker of DNA damage, and what we observed is that those mice typically have more endogenous DNA damage than wild-type mice, suggesting that there's some sort of persistent and endogenous damage that are present in those brain cells, which could explain potentially some of the development issue and also some of the neurocognitive defect that we see in the patient. So that's kind of the conclusion of that part. We are not denying any of what has been published before about the transcription regulation of POGZ and its importance. We just think that maybe it's a more complex disease than originally anticipated, where there's actually several elements of POGZ that are important, including the fact that maybe, POGZ may actually be involved in genome stability and could actually regulate DNA repair in this context. So that's kind of the work we've been doing in relating to the White-Sutton syndrome. Just to finish the presentation, I want to go to another study that we just published, where we look at the role of POGZ in cancer. And the reason why we went into that area, I'm going to go fast, I'm going to do my best. The reason why we went for there is because I think there's a mention at some point from Josh about the comorbidity, and our code those elements to actually be used as a way to actually raise awareness of a gene in terms of different disease. And as cancer is one of the key disease that we all experience, and trying to actually put awareness of the role of POGZ in cancer may be a way for us to draw funding to actually support work in the White-Sutton syndrome ultimately. And so that's, again, the same work that John continued to do that, and that's been in collaboration with Josie Siegel, who is an expert in triple-negative breast cancer. So I'm going to do very fast just to show you a bit of data. What we looked at, and what is very interesting actually, is that in patients with cancer, we see very rarely mutation in POGZ, unlike what we see in patients with the White-Sutton syndrome. However, what we do see is a dysregulation and an overexpression of POGZ. And so we look at POGZ expression in different types of cancer, and what was very striking is one of the top overexpressing cancer type was actually breast cancer. So we further look into that. You can see if you compare normal mammary tissue versus breast cancer tissue, there's a significant overexpression of POGZ in cancer tissue, tumor cells. you may or may not know, but actually breast cancer can be subdivided in different type of cancer or different type of subtype. The most aggressive is called triple negative breast cancer. And what we saw is that in that context, POGZ is down-regulated compared to other type of breast cancer. And importantly, the low expression of POGZ in those patients correlate with a poor prognostic, suggesting that if you have low expression of POGZ, you're more likely to die faster from breast cancer, which was very interesting and very striking to us. We kind of screen for expression of POGZ in different type of cell lines, and what we observe is that several breast cancer tissue, including basal and triple negative, actually express high level of POGZ, and one of them is actually a very good model to mimic a triple negative breast cancer. It's called the 41 model, where you can actually introduce tumor cells in the mammary fat pad and look at how the tumor develop and how it can metastasize. And what we saw is that POGZ knockout mice, or POGZ knockout cells injected, those mice actually have a growth defect compared to wild-type mice. And this is not the case when we overexpress POGZ. That suggests that POGZ is important for tumor growth. The other thing that we observe is that when we look at whether the tumor actually can metastasize to different tissue, we saw that POGZ actually knockout mice have actually an increased capacity to form metastases in the lung, which suggests that POGZ is important for the aggressiveness of the tumor. Let me just go over everything because I think I'm over it anyway. I'm going to finish here to make it simple. So I think the only thing I want to mention is that we think that POGZ may have a role behind the transcriptional regulation in a White-Sutton syndrome, which may be linked to genome stability. We also do have evidence that POGZ may have an important role in other type of pathology in cancer, primarily in the context of triple negative breast cancer, where we think that it may actually be important for the growth of the tumor, but also of the metastatic potential of those tumors. And in that context, we think that is a transcriptional role that is important for this element. And we are trying to propose, and that's some of the grant that I'm submitting right now, to get funding, that POGZ could be used as a prognostic biomarker for triple negative breast cancer and could also be used to actually prioritize patients for very specific targeted cancer therapy that we've been using. And this is a teamwork, so this is all the people that have been contributing to this work here and all the funding agencies that typically are coming from Canada. So far, I have not been able to secure yet funding in the US, so that's my process right now. But thanks to them, we've been able to do all the work that I've been showing you today, and hopefully we can continue working on that in the future. And I welcome any question from you.
Thank you very much. So, questions? Okay.
Hi, maybe a stupid question. I'm not an expert in this disease. Is there net accumulation of DNA damage in these patients?
It's a fantastic question. We don't know. So we have not yet dealt with any patient samples, so we don't know whether constitutively those patients have damage, but that's something we're very interested to look into. But
follow-up question. What happens to the expression of ATM?
I don't know, but that's kind of what we're thinking about as a target. So you know that there's ATM inhibitors that could prevent the signaling of DNA damage. And so we're thinking that maybe if you could prevent the signaling of those damage, you could actually also prevent the development issue or the growth defect that you may see in those patients by allowing to grow, actually.
Okay. Last question, I promise.
No, don't worry.
Is there telomere attrition in these patients?
Fantastic question. I cannot tell you, but I don't think people have looked at it, but I think it's something we should look into. Yes, definitely.
Okay, thank you.
You are welcome.
Hi, great talk, and I'm actually a fan of your student's work.
Well,
Yeah,
I'm a fan of his work too, to be fair.
And so I was just like wondering, is there like a hotspot or like specific location in the genome that you can see where there's accumulation of these like double-stranded break? Are they more in like the heterochromatin region? And I guess as a follow-up, or never mind, just that one.
So we haven't looked at that systematically. And I think that's something we should definitely investigate, you know. I mean, you may expect that in heterochromatin, you may have obviously a higher accumulation of damage based on the role of POGZ, but that's not something that you have correlated yet, I guess.
And I guess one more question is like, can you quantify essentially like the types of repair at a particular locus in the DNA? Like what type of repair mechanism is
Yeah, so I mean, we could ultimately, if you generate a very localized damage, to see which pathway is actually preferred in different contexts, including whether POGZ is present or not. Yeah. So we could definitely look into that, yes. But
any other question? There's a question. Go ahead. Okay. It's either one or the other.
Last one from Dr. Slatcher. She says, great talk as well. You suggested the POGZ's role in DNA breakdown and repair. Could it explain neural developmental function, but how do you explain the function of HDR in post-replicative neurons?
So let me reformulate what I said, okay? I think that it could be partially explaining. I don't think that is fully explaining the role in neuronal development. But, so the question was about, you saying about
How do you explain the function of HDR in post-replicative neurons?
Well, there is no HDR in post-replicative neuron because they're not dividing anymore. So it's primarily NHEJ that is using in those neurons, unless I'm mistaken. But so I'm not sure.
Sure, I could always tell her to follow up as well if she had
to
any
look into that for sure. Thank you so much.
Just a more practical question for parents relating to the DNA repair part of things. As you know, parents of kids who have POGZ, deficiencies, should they be more wary of things that can cause DNA damage, like X-rays?
It's a good question. UV, for example, maybe exposing less. I mean, I think naturally you should not expose your kids and yourself to any UV skin things, But I think that's something you may want to consider, I don't know. I think the problem is that we have no long-term perspective yet about what happened with patients with the White-Sutton syndrome because it's so recently discovered. So, but maybe on the long term, it's something that we need to be cautious about, I think.
Thank you.
Just as precaution.
Yeah, so Alex, I like the result that you're seeing in the triple-negative breast cancer. It kind of makes sense because triple-negative breast cancer has very low homologous recombination, so it may be compensating by increasing POGZ levels. So that actually makes a lot of sense. But I would expect those tumors that have high POGZ expression, the tumors to be more aggressive, but you're actually finding that it's the opposite, that low levels of POGZ seem to confer
metastatic potential. So I think, and that's where I think is very novel what we're proposing, is that you could decouple the primary tumor growth from the metastatic potential, which I think it's interesting. We actually see that if you overexpress POGZ, you form less lung metastatic tumor. Yeah. So it's clearly a transcriptional control of so what we think is this EMT transcriptional control of POGZ in those tumors, and that explains why actually they have less metastatic potential when they have high POGZ. And it's not related to DNA damage, because we see very actually limited DNA damage in the tumor in the POGZ knockout cells.
So I gathered from your presentation that our kids have trouble with their DNA cell damage. Are there any treatments or therapeutics that you're aware of that would help with the DNA cell repair?
So there are few, I mean, so normally, to be fair, what we try to do for cancer treatment is to actually do the same process that your kids had, which is to prevent the repair so cancer cell can die. In your context, what we would like to do is to stimulate DNA repair to compensate for that. So there are molecules that have been used that have been developed or under investigation to kind of stimulate DNA repair at a certain capacity, particularly the HR pathway, that are in clinical trial for cancer biology. So I think on the long term, that's maybe something that could be explored. If there's, I think what we're missing in our data, and I think I want to be very honest, I'm not trying to oversell anything, you know, that's very preliminary, is the patient data. We don't have so far any patient data that showed that actually there is more damage in your kids in terms of DNA. So I think that's something that we're missing and we need to prove that. But if that's true, assuming that's true, I think there's way potentially to stimulate those DNA repair pathway. Or what I'm thinking also is that the idea is that when there's damage, cells stop dividing. So my hypothesis is that when your kids have damage, or when they start growing, they have damage and therefore they stop growing, they stop developing their neurons, they stop developing their brain. And if you find a way to prevent that pathway to stop, maybe there's a way to bypass this issue. So there's two different angles. There's either to stimulate the DNA repair or to prevent the cell from stopping when there's damage.
And if it's already stopped, is there a way to?
Yes, absolutely. There's a way to actually recycle them potentially, yes. So, okay, I'm sure. Thank you.